I would like to introduce Laura Heiser, who for our today's webinar, Data Links, Data Science Research webinar, who will talk about research and data generated by Data and Signature Generation Center at Oregon Health and Science University. Please, Laura, go ahead.
All right. Thank you, Mario. What I would really like to focus on is not so much what we are doing for links, but what we have done over the past several years to get us where we are and leave us well positioned for really starting the next phase of the experiments that we're doing in the lab. And much of our work actually has been informed by work of the TCGA project. This project has been going on now for about 10 years, and over the course of 10 years has profiled cancers of many different tumor types across many different molecular levels, from genomic all the way up to proteomic levels, and as well there's even a bit of clinical data inside TCGA. And these studies have pointed out both the variation across the disease, across tissue types, as well as the variation in the disease within an individual tumor, or within an individual type of tumor, that is. And with this set of data in mind, I think the whole community has a much better idea of what the molecular changes are that are associated with cancer, and leaves us now well positioned to try to think about the biological implications of some of these observations. And that's really has been a focus of our work for a number of years. And in particular, what we have been doing is focusing on breast cancer. And so just what I wanted to slide was to enumerate some of the things that we've learned from TCGA, and where I think we still some little factoids that I think are sort of lead to open questions about exactly how this disease is being initiated and progressing in patients. And what TCGA showed us in breast cancer is that there are about 45 regions of recurrent amplification or deletion, about 10 distinct subtypes, each with a unique collection of genomic and epigenomic alterations. Of course, the number of subtypes observed varies depending upon the particular profiling data set being analyzed, and precisely the algorithm as well being used. So anywhere from sort of 5 as the classic, sort of somewhat classic case of the number of subtypes, up to 10. There are other studies that have shown that within the subtype of triple negative, there may actually be finer distinctions that one can make. And the importance of this is that as we begin to slice up the pie into smaller and smaller pieces, we think we can begin to really gain a foothold into what's driving particular sets of tumors to behave as they do. Additionally, coming out from TCGA was the observation that there are a few genes that are recurrently mutated, and many hundreds more that are mutated in just a handful of patients. There's also a fair degree of high inter- and intra-tumor heterogeneity, which has implications both for advancement of the disease as well as treatment of the disease. And therapeutic resistance is associated with increased aberration frequency. So all of these observations from TCGA, as I mentioned, sort of lead to additional questions about what do we do now that we know all of this? How do we best approach trying to understand it, and then trying to manage the disease and best treat the disease as well. And so to do that, we and others have put forth the idea that cell line model systems are a very effective way of trying to understand the biological underpinnings of different types of samples, in particular cancer samples. And this goes back now to sort of the links roots, which is from some of the work that Todd did when he first set up the connectivity map, to use a few different cell lines to try to understand response to therapy. But in the recent past, as many of you know and may have even been involved in, there have been several other groups that have put together large collections of in vitro cell line models. So for instance, we have the CCLE group. There's also a group out of Sanger trying to really collect different sets of cancer cell lines to profile them deeply and understand the biology of the disease. In addition, one of the benefits of looking at cell lines is we can look both at how they respond to therapy and also look at how they may be perturbed in other ways, such as done by the Project Achilles looking at the effects of siRNA knockdown. And just to then be very clear about precisely what we mean when we say we're looking at cell line models. And I've schematized that in the figure here on the upper left. And the cell line models represent tumors that have been biopsied from individual patients, taken into the lab, and then grown up in a dish. And these can now be cultured effectively indefinitely. And they then act as a renewable resource. They can be very well characterized. And it's also possible, as I mentioned on the previous slide, to take these models, these cell line models, and perturb them with therapy and ask how well they respond. And that in particular has been a big focus of the work in our group here. We have put together a collection of about 80 different cell lines and have profiled them on just about every platform under the sun. I'm showing here schematically some of what we've done. So if going around the ring, starting up at about 1 o'clock, we have RNAseq profiles on these cell lines, mutational profiles from Exome-seq. We have sort of the more old-fashioned array-based studies, such as the exon arrays, promoter methylation arrays. Down at 6 o'clock, we have 2 different sets of protein data. We have RPPA, reverse phase protein lysate arrays run by Gordon Mills at MD Anderson. And Gordon is also a part of our link center and will be contributing as well, which I will get to at the end of the slide. Additionally, done in a more low throughput manner, we have a pretty large collection of Western blot data on these cell lines as well. We also have copy number data coming from SNP6 arrays, which is still the most widely used platform for copy number. And finally, we, as I mentioned, have a fair bit of drug response data on this panel of cell lines. Almost all of these data at this point have been deposited into the public domain in one way, in one place or another. And I will give you some links to some of the easiest places to access them at the end of the slide deck. Just to be pretty explicit about the cell lines in our collection, as I mentioned, there's about 80. Not all of them have been profiled on all of the different platforms, which is simply because we've been collecting these profiling data over about 10 years or so. But this lists some of the core set of cell lines in our collection that we've been studying. And now what I would like to do in a few slides is to just give some overviews of some of what we have observed in these data. So we have, from SNP6, we have been able to get a pretty detailed look at the copy number profile. And that's what I'm showing you here in this figure, where this is a population level average from across a set of about 50 or so of the breast cancer cell lines, where we're looking at, along the x-axis, genomic position from chromosome 1 up to chromosome X. And on the y-axis, we are looking at p-values indicating whether or not there is an amplification or a deletion at that particular genomic locus, where amplifications are shown in red and deletions are shown in green. And what's pretty clear is that there are a fair number of regions of the genome that are recurrently amplified or deleted. So in particular, we see amplifications at NIC, at cyclin D1, and at ERBB2. And these I point out because these have been also very well described in the primary patient data, which gives us supports the idea that in ensemble, the collection of cell lines is a reasonable model system for a set of patient samples with a fair bit of data to support that point. Additionally, if we look at the gene expression data, in this particular case, the heat map shows RNA-seq expression, we can recapitulate some of the major clinically relevant subtypes. So in this particular heat map, each column represents data from a single cell line. Each row represents data from an individual gene. What are selected out here are a few hundred of these highly variant genes. If we use unsupervised clustering approaches, we can, as I mentioned, identify clinically relevant subtypes, including Claudin-low, basal, and luminal-like subtypes. Each of these subtypes has clinical implications, both for treatment as well as for prognosis of the disease. In the RNA-seq data, it's possible to look both at gene-level summaries, as was on the previous slide, but also to begin to think about alternative splicing. And this is work that was largely done by O.B. Griffith when he was a postdoc in the lab. He did a fair bit of work on alternative expression and has several, actually 100 example cases of genes that are not differentially expressed across the panel, but where once we start looking at the alternative expression of these genes, we do start seeing something that is subtype specific. And the example here is from the gene FRMD4A, which is a firm domain containing protein A, involved in regulating the cytoskeleton. So really the point of putting on an example like this is to simply to remind everyone that the data here can be mined many different ways. It's a really very, very rich data set that can be used for a lot of different studies, both to ask very specific questions about a gene of interest or a cell line of interest, or to do a more discovery-based genome-wide type of study. We also have Exome-seq data on the cell lines, which we've analyzed to identify mutations. And what we can do to a first approximation is recapitulate the handful of mutations that are frequently mutated in the TCGA breast cohort come up as also frequently mutated in the breast cancer cell line panel. So most notably, P53, PI3 kinase are two of the heavy hitters, so to speak, in the mutational space in breast. Of particular interest is to think about not just individual genes, but also to think about the pathways that are deregulated across this set of samples. And one approach that we have taken for this is to use the paradigm modeling approach, and this was developed by Josh Stewart's lab at UC Santa Cruz. And in brief, this particular algorithm takes the molecular profiling data, the baseline profiling data from copy number and gene expression, and marries that with information about curated pathways, so information from KEGG, BioCarta, and so on. And then using a Bayesian inference approach can end up yielding inferred pathway activity levels across each of these samples. And what we can end up doing is asking questions like, what is the relative expression of the PI3 kinase pathway in the basal versus all other cell lines? And what we have found is that it's actually a pretty useful way of trying to integrating across these data sets and trying to identify different pathways and hubs that are deregulated across subsets of the samples. Just a couple of additional vignettes on these. What we have done is to look at subtype-specific networks and pull out different pathways that are preferentially deregulated across particular subtypes, and are now actually thinking about how what are the implications in particular of the pathways that are less well known, and thinking about bringing in as well some of the drug target information into these sort of pathway maps to try to understand how you might interpret these deregulated pathways and whether they may end up informing response to particular therapy. As I have mentioned, that part of the theme of our studies has been to always map back to TCGA or some other source of primary data when we're analyzing the cell line data. And from the paradigm network inference scores, if we compare the scores from the cell line to the scores observed in TCGA, we find that we get pretty good agreement in the types of pathways that are deregulated across these two different cohorts. And in fact, we can pretty convincingly recapitulate a much stronger association with subtype than with the tumor or cell line of the site of origin, for instance, so to speak, which again gives us some confidence that we're recapitulating important biology in the cell line sample. All of that baseline profiling work sort of sets the stage for one of the key questions that we've been asking, which is how do we best predict who will respond, which patient will respond to a particular therapy? So how do we improve therapeutic outcomes for patients? And what we hypothesize is that we can use this now very well characterized cell line system to start to gain insights into that question. And that's sort of schematized here on what the concept is, which is to take this collection of breast cancer cell lines and use all of the baseline molecular profiling data showed schematically on the lower portion of that of the slide. And as well, we have a collection of drug response data. And so then to use various machine learning and statistical approaches to try to identify relationships between the drug response data and the molecular data to identify signatures of response. And it's worth spending a few minutes just describing what the drug response data are that we have. As I mentioned, we have about 70 different cell lines that have been screened against about 90 therapeutic compounds. And these compounds include small molecules, pathway-targeted therapies, and also chemotherapeutic compounds. And they are composed both of FDA-approved and investigational compounds. And the way the assay runs is to test 9 different concentrations in triplicate following 3 days of treatment, which is shown schematically by the little time bar on the left-hand side. So we grow cells up in the lab, plate them. 24 hours later, we apply drug. And this is then, drug is applied at one of 9 concentrations. And then 3 days later, we measure cell viability. And from this particular assay, what we can do is to generate growth inhibition curves, which is shown on the right-hand side. So what's plotted there is along the x-axis, we have drug concentration. Along the y-axis, we have growth inhibition, or the fraction of cells that are still living after 3 days of treatment. What we can do is then fit a sigmoidal curve to these data to end up with sort of the classic drug response, and then extrapolate out a GI50 value, or a concentration at which we inhibit growth by 50%. So this single metric has been the primary focus of our machine learning approaches, but it is by no means the only metric one could use from, could study from these different curves. And in fact, in other studies, we've been now exploring different aspects of this curve to think about what sorts of information we get out from there. And this actually was work done in collaboration with Peter Sorger. But for the purposes of these particular studies, we're focused principally on GI50. And just to give a little picture of what some of the compounds are in the and what they target in our collection. So we're targeting many of the major signaling pathways that have been well described and we know to be relevant in cancer, so P53, MAP kinase, JAK-STAT, and so on. There is admittedly a bit of a biased representation in some of these in the distribution in that, for instance, there are a lot of compounds that target PI3 kinase. And this is in part just because we have had a collaborator who was very interested in PI3 kinase, and so we searched that space pretty deeply. Here's a high-level view of those drug response data. On the left-hand side is a long string of box plots, where within each box plot are data. Now this is log transformed GI50 data, so drug sensitivity data for one compound. And what you can see is that the distribution in the response values varies from compound to compound. Some have a relatively narrow GI50 range. Others near the bottom have actually a very wide dynamic. Is everyone back? On the right hand, what I'm showing is a heat map of those same data, where each row represents data from a single compound, each column represents GI50, log transformed GI50 for a particular cell line. And we can cluster these data to identify patterns both in the way the cell lines cluster and in the way the compounds cluster in terms of thinking about their mode of action. What we've done over the course of several years is to really take sets of these compounds and to try to understand why particular cell lines are responding as they do. On this slide here, I'm showing the same type of data in the heat map. So here each again represents data from a single compound. Each column is data from a cell line. If we have studied a core set of about 90 compounds or so, and what we found is that about a third of them show subtype specificity, which is to say if we compare, if we simply ask whether or not subtype can inform whether or not a cell line is sensitive or resistant, then we say that it does. There's some sort of subtype specificity. If we take those subtype-specific compounds and cluster them, what we see is that the compounds cluster roughly by their mode of action. So, for instance, we see aurora kinase inhibitors clustering together EGFR and HER2 compounds next down this list, and so on. We see the PI3 kinase and mTOR inhibitors, which both target the same pathway, show similar sensitivity profiles across the panel of cell lines. We have also looked for associations between recurrent amplifications or deletions and drug response, and that's shown here on the right. And about a quarter of the compounds, where we did this analysis, about a quarter of the compounds showed some sort of association with recurrent amplification. So, for instance, response to the aurora kinase inhibitor GSK 1070916, the first box plot there, was associated with amplification at aurora kinase, at 20q13, which encodes aurora kinase A. And again, just to be explicit about how to interpret those figures, within each of these figures are data from one compound, the GSK 107 compound, where we have now stratified the cell lines based on whether they're amplified at aurora kinase or normal at aurora kinase, so either yellow or orange, and then ask, do we see a difference in their drug sensitivity? And in the case of this GSK compound, for carboplatin, we see an association with amplification at cyclin B1, and then not surprisingly, for compounds that target ERBB2, we see an association with amplification at ERBB2. Not all of those compounds in the lower box plot target HER2, but they target either HER2 or proteins that are close in the pathway to HER2. In addition to these relatively simple statistical approaches, what we've also done is to use some more sophisticated machine learning approaches to try to identify signatures of response from the baseline profiling data. And what we found is that we can identify robust predictors for about 50% of compounds. And that's shown here on the right-hand side. It is the case that for about a third of the compounds where we can identify a robust predictor, it is the case that that predictor also encodes the subtype information, the transcriptional subtype information. And so subtype alone would actually lead you to almost the same prediction of drug sensitivity as would the more complex multi-featured molecular profile. So those are the compounds that are shown on the lower half of the figure. Within each of these figures, or each of the bars on the right-hand side, each bar represents data from a single compound, where the height of the bar represents the AUC, so the area under the curve, for the predictor, where the gray portion of the bar is the predictor based just on subtype, and the entire height of the bar, the gray plus the turquoise, is the AUC that you can achieve if you use the entire feature set for generating a predictor, so the whole set of molecular features. What we have also asked is whether some of the predictive signatures that we have identified are present in primary data, so mapping back again to the TCGA data set. And what we found is that for about 22 of these compounds, it looks like there is evidence that the signature we observe in the cell line data would translate forward into the TCGA data set. And that's shown here on the figure. I won't actually go into the details here. In addition to doing all of these analyses within the lab, what we also have done is to become quite active members of the DREAM consortium. I know that many others in LINCS are also both aware of the effort and involved. But all of the data that I just described up to this point were the basis of the DREAM7 drug sensitivity prediction challenge. And for those who are not familiar with the DREAM, I will just spend a couple minutes telling you what it is. DREAM is the Dialogue for Reverse Engineering Assessments and Methods. And the goal of DREAM is to catalyze the interaction between experiment and theory in systems biology. And really, very important, is to build a multidisciplinary community around a compelling biological problem. So really to bring people together, working together and beginning to speak the same language and really think deeply about a critical and important biological problem. And this crowdsourcing effort was started by Andrea Califano and Gustavo Stolovitsky. For the past about 2 years ago or so, there was a merging of DREAM with Sage Bionetworks, and now all of the DREAM challenges are run through the Sage Synapse platform. And the Synapse group is additionally a very active part of our LINCS Center here, and has a lot of interesting resources to bring to bear, both to the problem of sharing data, but also putting together these challenges, because the infrastructure behind the scenes on one of these challenges is really quite remarkable for how much effort is required on the back end to keep all of the information organized and develop scoring scripts that can run automagically. So just in brief, the way a DREAM challenge works is that a consortium of organizers gets together and thinks about what would be a compelling biological problem to have the community engage upon, and then they either go out and try to identify relevant data sets, or to spend time actually generating data sets now that could be used for a challenge. I think this is a really important part about how the challenges are now being thought about. It's not so much what can you do with what already exists, but what are the data that you would need to generate in order to run a really effective challenge that could be that would be very compelling to the community and also would really advance the field. But once identified, the data set are split into a training set and a testing set. The training set is released to the community, and there's an open challenge phase that lasts about 3 months, although this is becoming a bit flexible now in the way the challenges are evolving, and I think sometimes it's both a longer duration as well as has multiple phases of being open. Once the challenge closes, participants submit predictions on the problem. Those predictions are scored against the held-out test data set. And the key really to the success of DREAM is the fact that the training set and the test set are very distinct, and that the test set has been held back completely from the training phase. So the particular challenge that we did was really to take these same sorts of problem that we have been working on to try to identify predictors of response and to pose that to the community. So what we did was to take 53 of our breast cancer cell lines and all of the baseline profiling data sets for them, that's shown on the left-hand part of the figure, and then for a set of 31 compounds where these drugs had never been released to the community anywhere, what we did was split those into a set of, or rather we took the cell line data and split that into a training set of 35 cell lines and a test set of 18 cell lines, where the data for the test set were held back and used for scoring. Participants then used these two data sets to identify predictors of drug response, submitted ranked list of cell lines, which we then scored the test data set. The drug response data we used, I've shown you here down at the bottom. In all in this challenge, we had about 44 teams from around the world participate, which was pretty remarkable, actually, to be able to take what we have been doing in the lab and multiply it into this huge amount of work that the community was very actively engaged in. All right. So the challenge itself was to predict the rank order sensitivity of the cell line in the held-out test data set. And once those ranked order lists were submitted, then that's what we used for scoring participants' predictions. And to actually score, what we did was to use a Concordance Index to score the different predictions. And I have a couple of slides that detail how this approach works. And what we do is to compute the Concordance Index, we build a list of all observed pairs of cell line responses and then test whether the predicted rank is concordant with the observed rank from the gold standard. And concordant would give you a score of 1, anti-concordant would give you a score of 0. So in this particular sort of toy case, if we have 4 cell lines ranked 1, 2, 3, 4, if the gold standard values were A, C, D, and B, and if we had a participant submit that the order should be C, D, A, B, then we would say, looking at the pairs of A and C, that those were anti-concordant because they were in the wrong order. The prediction was C and then A, and it should have been A and then C. If instead we look at the pairs of C versus D, we see that they are in both the gold standard and the prediction. C comes before D. We would call this concordant, and you would get a score of 1 for this case. So we go through all pairs in this way to identify the concordance index across all of the different compounds and all of the different pairs of cell lines. So the schematic on the previous figure assumes that we know with 100% accuracy what the correct rank order should be. But in reality, we know that the drug response data are noisy, so what we did was to go through efforts to account for the variation in the drug response data and use that as well to incorporate that into the scoring algorithm. And actually, I will not go through this in great detail now. But based on the variation, both on the dynamic range and the variation in the drug response data, we then weight each of the compounds in order to really account for the fact that for some compounds there was better ability to predict drug response than in others. And that certain compounds then could be considered more scorable than others. So for instance, in the drug 1, the weighted average would end up being about 2.8 based on the dynamic range of the compound, or of the cell lines. For drug 2, we see there's a broader dynamic range, and so the weight would be 4.1. And for drug 3 and for drug 4, we would end up with weights of 1.3 or 3.1. Across 47 submissions, here is the distribution of those weighted probabilistic C-index scores for each of the teams. So the gray bars here represent the null distribution, centered around 0.5. So this is the case if it was a completely random prediction, that we would expect a concordance of 0.5. Each dot represents the score from one of the teams. The top scoring team is the blue dot on the farthest to the right. Both that and the second scoring team were statistically distinguishable from each other and from team number 3. Which gave us confidence that the algorithm used in each of these methods vastly outperformed any of the other algorithms used in this particular challenge. In addition to make sure that the scoring was robust, we also did some subsampling of the data. Here, schematic is a little cartoon of the drug response matrix, where we're looking at cell lines along the rows, drugs across the columns. It is the case that in the drug response data supplied, there were some missing values, which are shown in black. But if we take the drug response data, mask 10% of this randomly, and then rescore each team and do that 1,000 times, then we can end up with a distribution of scores for each of the teams. And this actually is what we use to compute the p-values to assess whether or not teams are distinguishable from one another. Here's the distribution of all teams. Again, the top performing team on the left-hand side in the red box was statistically distinguishable from team number 2 and so on, and team 2 from team 3. The boxes here are colored by the algorithm class. So whether, sort of generally, what was the approach used for the prediction. The top performing team called themselves Team Sin, and they used a novel approach for predicting drug response. They used a Bayesian multitask multiple kernel learning approach, which is summarized here on the schematic. But there were 4 key elements of this approach. One is multi-view learning, where the heterogeneous input data are integrated into a single model. Kernelized regression, which computes the outputs from similarities between the cell lines. Multitask learning, which shares information between the drugs, so really trying to leverage all of the drug response data together. And then finally, a Bayesian inference component to handle uncertainty in the data. Other key aspects of their method is that they really brought in external data as well in this prediction. So they went out into the Sanger drug response data and brought that in, and additionally brought pathway information into their learning algorithm as well. Here's a distribution of all the scores once we sort them by the algorithm used, where we have, and the red, each of these bars represents the score from an individual team, where the height of the bar is the weighted probabilistic concordance index, and they're sorted by method and then also by score. And really, if we look and try to find patterns based on the class of algorithm itself, we don't actually find any distinction in performance based on the algorithm alone. But what we did observe is that those teams that used prior knowledge tended to score more highly, and additionally, the nonlinear modeling nonlinear associations also tended to lead to better scores in predicting drug sensitivity. So those were really the take-home messages, and it's those sorts of ideas that we're continuing now to incorporate in our work as we proceed, both in continuing on with our drug prediction work and also thinking about some of the work we're going to be doing in links as well. So as I mentioned, all of the data that I described on the Cel Line panel has been released into the public domain in a few different manuscripts. In addition, what's done is to post the pre-processed and cleaned data on a Synapse wiki. So there's a single place to go as a clearinghouse for these data. And both the pre-processed data are there, as well as little README files that describe what the data are, and a few links to the original manuscripts, and then links out as well to Geo if people are interested in getting to the raw sequencing data. All right, so actually in the last couple minutes, what I just wanted to do was to bring us back a little bit to links. So all of the work that I described so far was really focusing on intrinsic molecular features and cell intrinsic biology. And what we're now starting to think about with links is how extrinsic cell biology affects individual cells. So, which is to say, really thinking about how signals from the extracellular space, the microenvironment, how those modulate cells and change both their transcriptional programs and also change their phenotype as well. So it may be that different molecules in the environment lead cells to proliferate or undergo apoptosis, undergo a change in differentiation, and so on. And what we're really now starting to do is to try to think about bridging this gap from the microenvironment through the intrinsic molecular network space and out to changes in cellular phenotype. And the rationale for this particular approach is that we know that individual cancer cells and tumors are actually very much subject to the signals coming in from their environment. So here's a figure from a recent Nature Reviews paper that sort of shows that schematically, the number of different types of signals that cancer cells are receiving all the time. And it is now, I think, as a community, we are, and we in particular as a lab, are really appreciating that this is a really important set of signals that we need to consider, both for thinking about what cells are doing in their native state, but also how might these different microenvironment signals affect their response to therapy. And so to that end, for links, what we have designed is a set of experiments that we think will help us to better understand the effects of the tumor microenvironment. And to do that, what we have developed is what we call a microenvironment microarray, where we can grow cells on, we can spot down onto individual glass slides different microenvironment perturbagens. So these are proteins that we've identified as being important in the tumor microenvironment. Spot those down on slides, wash cells over so they bind only to those spots, and then using an immunofluorescent imaging-based assay, we can assess about 9 different biological endpoints where these endpoints are surrogates of differentiation, senescence, and so on. In addition, what we will do, and this is in collaboration with Gordon Mills, is to run reverse phase protein arrays following treatment with a selected subset of microenvironment perturbations, and as well look at the transcriptional changes induced by individual microenvironment proteins. All of this will be done on 20 cell lines, where these are principally cancer cell lines and with a bias toward breast cancer, although we are in collaboration with the other link centers. We have identified a set of about 5 different cancer cell lines that will be tested, and those are in common across the centers and represent different tissue types as well. So once we have these data in hand, what we will be well positioned to do is to try to identify phenotypic signatures of microenvironment perturbation. So using some of the approaches, analysis approaches I described in the earlier parts of the talk, to try to look for associations between the phenotypic imaging-based endpoints and the transcriptional and proteomic readouts. And we imagine with these data sets we'll actually be able to run analyses in several different ways, looking for associations between the molecular profiles and the phenotypes. And with that, I will just acknowledge all of those who have been a part of our link center, as well as a few others who were key contributors on other parts of this project. This list, although now that I'm looking at this list, certainly the other contributors is not at all complete. In particular, Jim Costello and Gustavo Stolovitsky should be listed on this for the collaborative work we did on the DREAM challenge. And with that, I will end. Thank you.
Thank you, Laura. That was brilliant. Would anybody have any questions for Laura right now?
So this is Avi. I also want to say that it was a fantastic presentation. So 2 things that I'm thinking of is the drug combinations and resistance. Is that something that is also somehow in your future pipeline to look at?
Yes, so we have done a little bit in drug combination studies. As you know, those expand really quickly when trying to think about what are the combinations of tests. So we have done this a little bit, really in collaboration now with Rosie Sears, who is a professor here at OHSU, and really thinking about how therapy may modulate differentiation status, and then using that as a guide for thinking about how to combine different therapies, so to try to come up with some rational approach. The other set of experiments are those led by Jim Corcola here, who has been thinking about using these microenvironment microarrays, and at the same time that you have cells growing on these microenvironment arrays, where we have the microenvironment proteins printed, to then also add drug and ask, in the presence now of both drug and a microenvironment protein, do we see a change in drug sensitivity, a modulation of drug sensitivity? And Jim has a couple of examples that suggest, yes, we need to be considering these other signals. So those are sort of the two, and that then leads to this idea of combination therapy, because then it suggests that you would want to treat with not just that particular therapy, but then think about a therapy that may also modulate the microenvironment protein in question. So those are sort of the two angles we've been working on for thinking about combination therapy. It is definitely the case that that's, I think, a really, really important avenue to go down in terms of thinking about effective therapy. It's also really complicated.
Mm-hmm. Thank you.
Any other question? Okay, well, I guess we can conclude this case. Again, thank you, Laura, very much. That was really, really great. And I will post the recording of Laura's presentation if you would like to revisit some things that she said. And also I will post some of the links on the website so that you can access the data that Laura was talking about. That's all, folks. Thank you. Hope to hear you next week.